Huperzine A is an alkaloid compound extracted from a club moss called Huperzine serrata. It's been touted to have numerous neuroprotective and nootropic effects, and it's been suggested as a possible treatment for Alzheimer's disease. Huperzine A is an alkaloid compound extracted from the club moss. The club moss has traditionally been used in Chinese medicine for fever, inflammation, and even schizophrenia. Because of its long history of use in traditional medicine, Huperzine A has been studied as a compound of interest for its potential effects on a number of important systems in the body and the brain. Huperzine A has been reported to be a potent, reversible, and a specific inhibitor of the enzyme acetylcholinesterase. This enzyme breaks down the major neurotransmitter acetylcholine, suggesting that some of Huperzine A's effects may come from generally increasing the levels of acetylcholine throughout the brain. For example, according to one animal study, Huperzine A was reported to increase acetylcholine level in the rats' brains for up to 6 hours after the administration. Several animal studies have reported that Huperzine A may result in more prolonged increases in acetylcholine in the whole brain when compared to other compounds that raise acetylcholine levels. According to one study, Huperzine A worked in a similar way to some Alzheimer's disease medications, which are believed to affect the acetylcholine system in the brain. The effects of the Huperzine A were seen to be more favourable and had less side effects than the prescription medicine. The potential increase in acetylcholine levels seems to differ between different parts of the rat's brain. According to a few animal studies, following the administration, the maximum acetylcholine level was observed in the hippocampus after 30 minutes, and in the frontal and prefrontal cortex after 60 minutes. These observations suggest that Huperzine A may influence the function of different parts of the brain at different times. However, some research has also stressed that the Huperzine A likely also affects other brain mechanisms that don't involve the acetylcholine, so this effect alone is probably not the whole story. It increases norepinephrine and dopamine, but not serotonin. This is according to one animal study. Early evidence from a few studies suggests that Huperzine A may have a few mechanisms that could help to protect the brain from stress and harm. Some of these suggestive findings include muscarinic and GABA receptors that can play a role in some of the reports of the Huperzine A protecting against seizures. One literature review on Huperzine A gives a hypothesis that it may help to promote neuronal growth by stimulating the production and the secretion of nerve growth factor. One in vitro cell study proposed that the Huperzine A may help to prevent or to reduce cell damage from the lack of oxygen or hypoxia. This protective effect may be partly mediated by the cholinergic anti-inflammatory pathway through alpha-7 nicotinic acetylcholine receptors. One animal study has reported that Huperzine A may help with inflammation in rats, in particular by reducing the activity of NF-kappa B signaling, and this may happen both through the inhibition of acetylcholine esterase or in other ways. In a systematic review of different interventions for Alzheimer's, combined data from multiple studies was used to suggest that Huperzine A may have a statistically significant effect in reducing cognitive decline during Alzheimer's disease. However, while the early results are promising, there are some important limitations to note. For example, the quality of these studies is somewhat lower than the quality of studies of other medicinal interventions in Alzheimer's disease and other dementia-related conditions. According to some other researchers, Huperzine A has been reported to prevent neuron loss during Alzheimer's, specifically by inhibiting the inflammatory response caused by the buildup of amyloid beta, the main factor that's believed to be responsible for Alzheimer's disease. In a related study, Huperzine A was reported to inhibit the NF-kappa B pathway in immune cells in the brain. This further suggests that it may help to reduce inflammation, which could account for why Huperzine A has been claimed to help to slow down the progression of cognitive symptoms in Alzheimer's disease. At least one clinical trial in patients with Alzheimer's disease has reported that Huperzine A supplementation appeared to improve cognitive function and overall well-being in these patients. According to some early animal studies and clinical trials in humans, Huperzine A may be associated with enhancements in memory and other neuroprotective effects. For example, Huperzine A was reported to significantly improve memory abilities in adolescent high school students when compared to an inactive placebo treatment. Although the mechanisms behind this potential effect are still unclear, one animal study reported that Huperzine A may stimulate the growth of neuronal cells, or neurogenesis, in the hippocampus of the mice. This brain region is believed to be highly involved in memory. While primarily a mood disorder, depression can also involve disruption in cognitive ability, which can be a serious symptom in its own right. According to one preliminary study, when used as an add-on or complementary treatment to standard antidepressants, Huperzine A was reported to resolve some of the significant cognitive symptoms associated with depression. One early study based on self-administration of Huperzine A reported a decrease to the individual's perception for the effects of cocaine. The author of the study proposed that Huperzine A supplementation may be a safe, complementary approach to help to treat addiction, although more research would be needed to verify the potential use. Myasthenia gravis is an autoimmune disease where the immune system attacks acetylcholine receptors, leading to problems with voluntary movement and muscle control. It's most commonly treated with immunosuppressants and acetylcholine esterase inhibitors. However, some early studies have looked at Huperzine A as a potential additional form of treatment. According to one preliminary study on 128 patients, the Huperzine A was reported to alleviate the muscle weakness in the myasthenia gravis patients. Preliminary findings from some very early studies suggest that Huperzine A could have a number of mechanisms that may potentially help to protect brain cells from certain forms of stress and damage. Nerve gases and organophosphates are irreversible inhibitors of acetylcholinesterase. Some research has suggested that Huperzine A may aid in preventing the seizures and the nervous system dysfunction that's caused by soman, an organophosphate. Additionally, at least one animal study has also reported that Huperzine A may also help with chemically induced cognitive impairment in mice. Glutamate is an amino acid neurotransmitter, and it can kill neuronal cells by overstimulating the NMDA receptor, a process called excitotoxicity. This can be especially dangerous to older neurons. Huperzine A is an inhibitor, an antagonist of these NMDA receptors, and this has led some researchers to suggest that it may help to prevent the neurons from cell death due to the glutamate toxicity. In addition to binding to the NMDA receptor, some evidence suggests that Huperzine A may also help to reduce oxidative damage from glutamate toxicity, specifically by activating brain-derived nootropic factor dependent and mTOR signaling pathways. Findings from one cell-based study suggest that Huperzine A may also protect against oxidative damage from iron overload. In addition to some of the preliminary evidence suggesting that Huperzine A may protect brain cells from certain types of damage, there's also a few early studies that suggest that Huperzine A may also help to protect the brain as a whole from certain forms of physical injury. For example, combination treatment with Huperzine A, together with other supplemental compounds like vinpocetine, acetyl-L-carnitine and N-acetylcysteine, alpha-lipoic acid and fish oil, have been reported to help to increase blood-brain flow and to improve cognitive function in retired NFL football players with a history of concussion. In one animal study, Huperzine A was reported to decrease the programmed cell death or the apoptosis, as well as improve neurological symptoms in rats with spinal cord trauma. According to one cell study, Huperzine A may aid in the recovery from brain bleeding by preventing damage to the mitochondria and postponing cell death. Although this evidence is very preliminary, at least one animal study suggests that Huperzine A may have the potential for protecting the brain during infections. In a rat model of severe bacterial infection or sepsis, Huperzine A was reported to protect the brain from damage by reducing inflammation. According to some early animal research, Huperzine A may be an effective treatment for epilepsy. In other animal studies, Huperzine A has been reported to to protect brain cells from overexcitement and death associated with convulsions. According to another study in mice, Huperzine A was reported to make certain brain circuits more resistant to seizures. According to some studies in humans, oral huperzine is absorbed relatively quickly, distributed widely throughout the body and the brain, and is gradually eliminated or removed from circulation at a moderate rate. According to an animal study, the level of Huperzine A were reported to be the highest in the kidneys and the liver 15 minutes after injection. After 12 hours, no trace of Huperzine A was found anywhere in the body. According to one human study, it was reported that a very high dose, 1 mg, caused peak serum blood concentration of the Huperzine A to be reached at approximately 80 minutes post-injection. Additionally, half of the Huperzine A was found after approximately 5 hours, suggesting that it may be necessary to take Huperzine A multiple times a day to achieve a steady, continuous level throughout the body. No build-up of tolerance or loss of biological sensitivity to the compound has been reported from repeated or continued use of Huperzine A, at least according to one study. Doses in the clinical trials for Alzheimer's range from 0.2 mg to 0.8 mg. However, for Alzheimer's patients, one study reported that a dose of 0.4 mg twice per day was required to produce clinically significant results, while 0.2 mg twice per day did not. In high school students who struggle with memory, only 0.1 mg was reported to be sufficient to produce detectable effects on memory ability. Early evidence available so far suggests that adverse side effects are relatively rare. For example, no notable side effects were observed at doses between 0.18 and 0.54 mg, at least according to one study on humans. However, some other studies have reported occasional mild side effects, including cholinergic symptoms such as dizziness, nausea, digestive upsets, headaches, and a decreased heart rate. One of the only available studies on the potential interactions report that Huperzine A is unlikely to interact with the cytochrome P450 enzyme, the enzyme involved in detoxifying drugs and other foreign compounds from the body. For more information about herbs, supplements, or natural treatments, check out my website.